I'm Ann Goldberg from Washington University Medical School in St. Louis, and we were moderating the late-breaking clinical trial sessions.
Hi everyone, Seth Martin from Johns Hopkins.
Hello, I'm Raul Santos from the University of São Paulo, Brazil.
So I think we had a terrific session with a lot of very good science. And the very first talk was a PCSK9 gene silencing therapy with very impressive LDL reductions with just 2 injections about 90 days apart, with LDLs going down to about 60%. Very impressive data from that presentation by Dr. Ray.
Yeah, I agree. I felt like that was an interesting approach where you would end up with only needing maybe 3 or so injections over the course of a year, and certainly that may be something that a patient would welcome.
Yeah, the results are very similar to what we have with the antibodies against the PCSK9, and apparently tolerability and safety was okay. It's interesting because with this kind of medication, actually is different from the monoclonals that bind PCSK9 into the plasma, and the monoclonals don't get into the tissues. Actually, this one goes directly into the liver. So one might wonder if we might have some kind of side effects of PCSK9 inhibition, because PCSK9 not only regulates the expression of the LDL receptor, but also has other functions in the body. So this kind of approach, I guess, would be very, very interesting to understand more. Apparently, they had no problems at all, right? Yeah.
So far, the safety signals look good, but certainly that's something to keep in mind moving forward, and will require further long-term data.
Yeah, bigger number of patients, and maybe in the future, maybe an endpoint trial, we don't know, right? But it was very interesting to see the results.
For a phase 1 study, yes, I think it was very interesting.
Yeah.
The other, I thought, very interesting data were the ANGPTL3 antisense with may have a different population of patients that would be targeted, and that was the antisense has been kind of tough in terms of side effects, but this particular approach didn't seem to have as many side effects as other antisense oligonucleotides.
Yeah, it's amazing how this technology is evolving. I have a big experience with mipomycin as the 2nd generation antisense for homozygous FH patients, and indeed, the majority of patients had injection site reactions, and most of the patients who dropped out from the studies actually had the flu-like symptoms. And apparently, we in this small trial, we didn't see any. And there's also another one with the same technology for Lp(a) that has been published recently on The Lancet that suggests that not only effectiveness, but also the tolerability of these newer techniques are ways of preparing the antisense. It's a lot bigger than the ones that we had in the past. So it's quite interesting. And actually, the population, as you said, and the target for this population, maybe patients with severe hypertriglyceridemia, mixed hyperlipidemia, and they might have benefits. And we have some genetic data suggesting that ANGPTL3 and also C3 that was also reduced by this antisense, ApoC3, might be associated with an increased risk of cardiovascular disease. So we might get some hopes for the future for patients with combined dyslipidemia or hypertriglyceridemia.
I think of course, the big headline, I think, is Glasgow, the Evolocumab study with the IVUS trial that Steve Nissen presented. And I was not really surprised that it actually worked, given that I'm a very LDL-centric person, but it was very nice to see that atheroma data presented.
Yeah, I agree. I mean, it's very exciting. We're dealing now with a therapy that's already approved. We're using it in patients. 2 of these monoclonal antibodies against PCSK9 have been out for over a year, and as we await the definitive outcome trials, it's really reassuring to see these IVUS data really strongly going in the right direction. It wasn't a questionable signal. These were highly significant results where we saw plaque reduction with PCSK9 inhibitor therapy. And I think, as Dr. Nissen pointed out, there's been a number of IVUS trials done before, and it is tempting to compare this study result with those prior study results. But it's important to treat each trial itself and look at the 2 comparison groups here. And clearly, the PCSK9 treated group had significant benefit when you're looking at atherosclerosis and regression. Yet there still are some progressors, and that'll have to be addressed in the future. But I was really encouraged. I think this keeps me very optimistic about this option for our patients, and and what I think I'll be absolutely overwhelmed with shock if the outcome trials don't come out in the right direction.
Yeah, and interesting, the results that we had in the trial actually were on top of statins, right? Actually,
Yes.
it wasn't a comparison against placebo by
Yes.
itself, right? So we had 60% of the patients taking high dose and 40% moderate dose statins. And the study clearly showed an additional benefit on top of the statin. Interesting, it's important to say that 40% of the patients who got the statins actually also had regression on the plaque, comparison with 67% with those who took Evolocumab and a statin. That's showing that statins are still good in doing their job. Of course, they're not perfect, but they still need to be used, right? So
they're setting a high bar, so
Yeah.
it's equally impressive
Yeah.
that we're seeing these strong results on top of statin therapy
Yeah,
with the
that's-
PCSK9 inhibitor.
And I
great.
think that the substantial LDL lowering really, I think, is very good for those of us in the field for a long time who felt that, lower is better, and that as we get lower and lower, we're seeing good results.
And apparently they got up to 20 milligrams per
Yeah,
deciliter of
Dr.
LDL.
Nissen.
They still
Yes.
saw the regression,
Yeah.
right? So this is very interesting.
Yeah, he showed that very straight line all the way down to LDL cholesterol levels of 20, and we're still seeing regression there. So I found that very interesting that it was still a linear relationship.
So the big issue then are the trials with the HDL mimetics, and two different studies, one a safety study and the other one pretty much taking ApoA1 Milano, at least that version of ApoA1 Milano, out of the running altogether. So I think that it's going to be very difficult in the very high intensity statin era to show additional benefit of modifying HDL. We still have a few things left that are being studied, but ApoA1 Milano certainly seems to not really be appropriate, and I thought that Dan Rader's analysis of why it's not really like the wild type ApoA1.
Yes, and interesting when we take a look at the population that has the ApoA1 Milano, and it was very similar what happened today, because usually they have lower HDL levels than the usual population, higher triglycerides, lower HDL, but in the end, of course, they have lots less cardiovascular disease in the long term in comparison with normal wild type people who has normal A1s. So, but even so, it's really sad, right? It's sad because we believe that HDL has to do with the atherosclerosis. It's hard to prove that nowadays with all the therapies that we have, that raising HDL or modifying HDL function or whatever will really prevent cardiovascular events. But I think we still need to go on. Interestingly, there are still other molecules that are being developed in the market. There's one study, very interesting study, that's being done by Syrenis. With patients who have genetic HDL deficiencies, and deficiencies in ApoA1 mainly. So these patients might get some benefit, because even taking statins, they still have lots of cardiovascular events. Mm-hmm. So let's wait and see. For special populations, maybe this kind of therapies might be good.
But at least the safety data with the CSL 112 suggests that at least that preparation was not harmful,
yep.
Has been a concern before. So certainly for a pilot study, it gives an idea. The thing about those data were that it's a little disturbing in a study with a few hundred people that the events, even though you can't say anything about events,
mm-hmm.
they weren't exactly trending in the right direction.
Yep.
Yep. Did you notice that too?
Yeah, I noticed. I guess I chalked it up to it being not powered for events, but it's as compared with our enthusiasm for PCSK9 and LDL lowering, it's frustrating here in the HDL field to see where we are after really decades of work in this area to still not have a proven therapy that benefits our patients and have that option, when we really, based on the totality of evidence, really think that this is an important molecule that's involved in cholesterol, reverse cholesterol transport, but also a bunch of other functions that we haven't been able to come up with a way that's effective to help our patients when it comes to the HDL pathway. So it's nice to see that work is still persisting here. There is still some lingering hope, but it's been frustrating to watch, and turned out, I think, to be more complex than any of us ever envisioned.
Sure. Epidemiology is not so clear, right?
No,
Low HDL, okay, it's still
yeah.
a marker of high
Right.
risk, but modifying it's another story, right?
HDL is complicated.
Yes, it is.
Yes, yes.
Yes.
I mean, I think that we're pretty happy with LDL, but HDL is complicated. We're going to need to wait for the other outcomes trials before we say for sure.
So
Okay, I guess we
I
discussed
think we
all
covered
of the
we
trials,
covered
right?
all the trials.
So I think we've
Yeah.
discussed, it was a very stimulating session, well attended. I think we had great
Yeah.
presenters, discussants, a lot of
Yes.
very good questions. And so I think that it was, we all enjoyed it very much.
Yeah, it was great to participate.
Yeah, it was a wonderful session. I really enjoyed participating. We had a packed room, standing room only, and I think a lot of enthusiasm for the future of the lipid field.
So I think that concludes our discussion, and I want to thank you and thank Seth and Raul for a very stimulating discussion.
No, actually it was a very nice session. I think the science advanced a lot, very important results, and I was very, very happy to participate in it.
I totally agree. Thank you very much, Ann and Raul. It was a pleasure.
Thank you.